Okay, so to give you a bit of background and a bit of introduction, so I work at the UK Dementia Research Institute, which is an institute that covers the UK. And as you can see, there are now 9 sites working together to do the basic and the translational research into neurodegenerative diseases and dementia. So how it started? Well, it started in around 2011 when UCL were fortunate to be awarded a grant of £20 million to push forward and enable research into neurodegeneration at UCL. At that time, there wasn't any focused biomarker studies. And so Professor Henrik Zetterberg was recruited from Gothenburg University to set up what would be the first kind of dedicated laboratory to look into this. And I joined the next year as a postdoc in the lab, where we initially were really concentrating on how to make the measures that we had for CSF, amyloid, for example, tau, better, because obviously these things always need to be improved. And at that time, we didn't think about blood because most people didn't think that we'd ever be able to measure proteins that would give us information about brain health in the blood. So that's where it started. And then a couple of years later, we have what we call a pivotal moment here. We actually got the opportunity, or it was brought to our attention by Quanterix, that they had this platform called the HD1, which was more sensitive, which would enable us to assay proteins with much more sensitivity than we'd been able to before. And as luck would have it, a Wellcome Trust grant opportunity came to us at the same time. And so we applied for this, and many people from the Institute put their support behind this. And we were fortunate enough to be actually awarded the money in 2015. And so this was indeed a pivotal moment. Now, this is a picture of the HD1 when we started. Unfortunately, the whole team are in front of it, so you can't really see it. But that is the only one I can find of where we were originally. So, moving on. But then what happened next was that we were able, or Henrik was able, to place a bid to the Dementia Research Institute, which was newly formed in 2017. And he was awarded the position of foundation group leader into the Dementia Research Institute, which was amazing for our lab because it meant that we could now move into new premises where we actually had some room and we could expand our group. And so the pictures there are basically where we came from, the horrible tall building on one side. In the middle is the cruciform building where we moved, which is beautiful. And there's one of the HD1 being set up on the day that we moved in. So it's just a skeleton there. So what we got when we moved into the cruciform building and joined the Dementia Research Institute was further funding. So we were able to buy another HD1 machine. And this gave us the ability to really start to collaborate with people to measure samples. They didn't know anyone else who had this bit of equipment, so people would send their samples to us and we would collaborate with them to do their projects. And so, yeah. And so this grew, and it became a bit unsustainable that we could just collaborate and provide all the materials for projects. So we decided that we needed to formalize what we did. And the Biomarker factory was set up. We actually spoke to the directors of the DRI the day before lockdown happened. So then we had plenty of time at home to sit down and hone our application to get the funding to become what we call the Biomarker factory. And so this enabled us to buy another HD1, or perhaps it was an HDX by then, and now have more kind of machines and equipment to work for the DRI and the wider academic community. So what happened then is that we moved back to the Institute of Neurology because we all ran out of space at the Cruciform building. And we now have 4 HDX machines in our space. And we, as I'm just going to tell you, collaborate really widely to do lots of projects. And I'm going to tell you about some of the publications and some of the stats for what we do. Okay, so let's look at the research in numbers. As you can see from the chart that we have here, is it? Yeah. Okay. Okay, so as you can see here, the majority of the studies that we've used have used the neurofilament light kit. And when that was first issued, it was a real kind of game changer because it's such a versatile and useful marker. And the only way to accurately measure in plasma or serum the neurofilament element of what we know is going on in the brain. But we've also used extensively the fourplex, the neurofourplex kits. We use the fourplex A and B in lots of TBI studies, and the fourplex E more in your Alzheimer's and dementia studies. And obviously, in the last 5 years, phosphorylated tau assays have become the kind of the main deal in Alzheimer's research. So today, our largest project is around 5,500 sample or 300 samples, and we measured that using 2 different kits. We've measured 5 projects so far with over 1,000 samples, and most projects contain between 100 and 500 samples. So publications to date. So this is only papers. This is only projects that have been published. We've got lots that are still ongoing, still being written up. But we can see that we do a lot of studies in AD, FTD, but we also cover the whole kind of gamut of neurodegenerative diseases. And of course, after 2020, we've done a number of COVID-19 studies and the consequences for neurological health. We also have worked on many one-off studies on diseases such as, well, CMT. We've done a couple of things on. We've done some HIV studies. And then we've also done a number of preclinical studies, i.e. we're able to measure neurofilament light in mouse samples really effectively. Neurofilament light is conserved across species, and actually in mice, and this is really handy, you can dilute it 40 times. So that means that actually we do get enough serum, we do get enough plasma to be able to make useful measurements in the mouse models. So that's useful. I know that recently we've also looked at phosphorylated tau in some humanized mouse models, but not had any data published yet, but it's possible to do using this platform. Okay, so looking at some early publications, we obviously adopted the system in 2015. It took a while, I'm going to say, to get going because a new technology can be viewed with some suspicion, and also a new technology that might have a cost attached to it. That's even harder, but we did get going eventually. And I think one of our first studies published was about plasma tau. Total tau was one of the first assays that was actually offered. And I can see the author in the audience looking at me. And so that study was FTD, where Martha and co showed that plasma tau is raised, but only in genetic forms of FTD that have the MAPT mutations. And these papers I'm only going to really briefly talk about because we could be here all day. So then we looked at neurofilament light, or a group at UCL looked at neurofilament light and total tau concentrations in CJD, and they showed that tau, but not neurofilament light, actually correlates with the rate of disease progression. Down syndrome was an early disease that was looked at using neurofilament light, and This was published in 2018, but we've seen many studies since showing how neurofilament light is associated with age and dementia status and stage of disease. And also, high levels of neurofilament light at baseline suggest often things about the disease, how quickly it's going to progress, etc. Now, we've got a strong Huntington's disease research element at UCL, and we've worked with them on a number of different studies. And here, this really nice study shows how plasma NFL really mirrors what we see in CSF. And also it it rises in pre-manifest HD, as you can see, and then it is highest in manifest HD. And we can also see that it correlates with it correl- plasma NFL correlates with CSF new NFL and is very much indicative of disease stage and status. So we've also done a number of interesting studies in familial Alzheimer's disease. Now, obviously, this is really useful because we can look at the genetics of the subjects here and know whether they're gene carriers or not, and look at neurofilament in longitudinal studies in estimated years to onset. And we can see from the longitudinal sampling graph here how the neurofilament levels take off when the disease is just about to kind of manifest. And so that's really been useful to see that, because obviously these diseases are ones that inform lots of the study on the sporadic forms. But when we also see that the neurofilament light levels correlate with neurodegeneration markers and kind of cognition markers, and cognition, and also tau and phospho-tau. Now, I talked about using neurofilament light in mouse models, and this is a particular study where we measured the neurofilament light to show that actually where a viral vector was used to replace a gene that had been knocked out in a mouse, we were able to show that neurofilament levels remained low where we were able to restore the function of this gene. And you can see in the chart B, the mock with the high NFL, that's where it's a mock viral vector, but where the proper viral vector's in, that's the full. You can see the much lower neurofilament light. We've actually done a number of studies now with this group, so we've got a few publications to look at, all regarding the Charcot-Marie-Tooth disease models in these mice. We've also done a lot of work with the TBI groups. We particularly collaborate with Dave Sharp and co at Imperial. And in this study was a BioAxe TBI study. And here, this was, I think it was about 2021 or 2. We looked at actual fourplex A. I think we started with fourplex A anyway, looking at how the markers change with time after injury. And also, we showed that you were able to use the microdialysate from inside the brain. They put like needles in the brain and took out samples and those measures of those are shown in blue on the chart under the brain. And we discovered A, we needed to dilute these samples many, many times, but B, Conteris had to make a new kit because actually the fourplex A, the GFAP in there didn't have a wide enough range. And because in neuronal TBI, the GFAP goes so high because of the trauma and the damage to the brain that they needed to make fourplex B, basically, which had a much wider range for the GFAP and stop that blowing out the rest of the results. So this paper, it looks nice now, but it was a bit of trial and error to get that to where it was when it came out. We also see that we can use neurofilament light as a marker of treatment efficacy. But crucially, this isn't always immediate. And a study done by our colleagues at the Institute of Child Health, they gave us the CSF NFL to measure, and it shows that actually in this enzyme replacement therapy for Batten's disease, we do get effects on neurofilament light. But this, as you can see from pretty better on the second chart, is actually up to 3 years post treatment. And so neurofilament light as a marker of efficacy or normalization isn't always quick. So you need to remember that. Also, one of our bigger studies that we've done to date was in collaboration with Imperial College and the UK Biobank, where we looked at, with Paul Matthews, people who had imaging and before COVID, and then imaging after having COVID. And so what they show here, and this is the C and D are pre-COVID, and then the middle two graphs are post-COVID, and the end two graphs are the change. And what we see here, and this perhaps isn't that surprising, is that these infections cause the markers of amyloid deposition to actually, or pathology to actually be worse or rise. And so that was one of the COVID studies that we did. More recently than that, we've worked with Adam at Oxford, who has been using cervical lymph fluid, or has been providing us with cervical lymph node fluid, so that we could investigate to see if we can measure these neurodegenerative biomarkers in this apparently less invasive fluid. I'm not so sure, but anyway. But we proved that we can measure them in this, and although we need to do a much bigger study, there is evidence of reduced brain lymphatic drainage with age of phosphatau 181. The significance of that, I think, perhaps needs a bit more investigation. We've also been involved in projects where we actually make assays in the lab, and the Simoa setup is very amenable to that, and you can get lots of help to do this also. And so Adrian Isaac's lab, who do a lot of work on FTD and various mutations that cause FTD, particularly C9orf72, set up this trial-ready assay to measure the GP dipeptide repeat protein that is found in CSF in patients who've got this mutation. And they're currently working on a number of different assays for this, or for different dipeptides. So for all of this, one of the things that we do work with lots of clinical researchers who are really interested in how the biomarkers that we use are going to be used in diagnostic settings. And Gemma Hazan, who's a clinical research fellow, is doing a number of studies with using phosphorylated tau 181. And she's been looking at whether or how the provision of a result to a clinician in a memory service changes their diagnosis or their confidence in their diagnosis. And she's also been looking at how plasma biomarkers actually reflect the CSF amyloid status. And I think actually we now know, and lots of people at this conference will be saying this, we know that they reflect them really, really well. So things we've learned in the last 10 years or so. One, working with new technology is a journey, and it can take some time to get it not get it working, but get it embedded into your lab and, into the things that you do, so that it's like second nature. We have found it's essential that we get regular training of users. And in fact, I banned people from using it because they break it. So it's really important that you don't make life harder for yourself by having people not knowing how to use it properly. We have regular meetings with our field application scientists and business development person to find out what's going on, what's new, what's going to be helpful for us, but also to let them know how we're doing so that they can learn from our experience as well. Maintenance is one of the most essential things here. We actually were gifted an HD1 machine very early on in this journey from Oxford because no one would look after it. And so they gave it to us because they knew that we would look after it and they could come and use it when they wanted. And so we learned really early on that you have to have a schedule for maintenance. And regular running of HD platforms is optimal. Our platforms, even though we've got 4, they all run a couple of times a week at least. And sometimes they're running all day, every day. And that's really how they like to work. And so just buying a machine to use it every now and again, it really not that. I don't see much point in that, even though maybe Conteris would like you to. The lab environment, it's actually more important than we thought at the start, i.e. the temperature of your lab and the water quality are really important considerations. So that's something to think about if you're going to start running these machines. We also have, after lots and lots of experience of people coming to us years after they've done a study to run another study and then try and compare it, we've now set up a system whereby we have the largest pool of QC samples we can have. I know it's not infinite, it will run out. But at the moment, if someone comes back to me and says, oh, I've just remembered I've got another 1,000 samples in that set I want to measure, we can QC it with the same samples we ran before. And that is something that I don't think I can stress how important that is enough. And which goes on to my next point, which is really plan your longitudinal projects. How are you going to bridge them if you need to? And also sometimes if we know in advance we're going to get multiple timelines, then we will make QCs out of that sample set. So you can do that. And then, right, I'm getting to the end now. Working with us. So it's always nice if we meet people who want to work with us, especially if they're just starting a project from the collection point, because the sample quality is really important and it makes sense. If you've got super sensitive technology, they're going to want super nice samples. So we've got lots of SOPs set up for processing and other aspects of your sample quality. We work with a service agreement that covers the MTA and expected roles, i.e. what we will do and what we expect you to do also. And then silly things like what we do with the samples at the end, how we destroy them. And the data we provide as requested. There's not many ways apart from an Excel sheet you can have it, but if you request it, we can do it. And then of course, manuscripts. We like to see manuscripts where we've provided that data because we can check that you've got the methods right, that you've got the CVs for the quality QCs right, and things like that. So yeah, and I think that working with us is a good thing to do. I should say that at the end. So, and then that's just the lab through the years, which is mainly Simoa related. And thank you. So thanks for asking me to speak today, but thanks also to everyone who funds us, collaborates with us, and gives us the samples. And obviously, thank you for listening.